Hello everyone, this is Maisam Ali and this is my progress report of my third semester. I am the student of Department of Life Sciences for Master Program. The name of my advisor is Professor Hongkong Tsai. This is my progress report checklist. The title of my research is the antimicrobial effect of far-UVC to 2 nanometer light therapy in Rat Corneal model. This is my third semester courses which I selected for my this semester, and this is a poster presentation in conference which is compulsory before graduation, and this is the bench work which I learned in my this semester. This is the study design for my current progress. In this study design I have three groups. This is a control group, and this is the traditional control group, and this is the experimental control group. Traditional control group means that this we just use iodine on the rat cornea, and the duration of the experiment is 6 hours. Objectives of my current progress research is to check, we already checked the safety assessment and disinfection ability of using far-UVC to 2 nanometer, and determine to assess the efficacy of far-UVC to 2 nanometer and their virulent effect on cornea. And disinfecting method of using far-UVC is better than using a traditional method of using iodine before surgery in the field of ophthalmology. This is the disinfection way of eye before surgery in the field of ophthalmology. Actually, we got the data that podium iodine have some side effects, and it make inflammation pain in eye before ophthalmic surgeries. So this is a result section. So this is a figure 1 in which I did the H&E staining, and we see here that the black arrow indicated the some pathological changes after exposure more than 5 seconds. And this is a significant figure which also show clearly. And this is the figure 2 in which we also check the ROS, rect oxygen species. If we give exposure to the cornea for more than 5 seconds, so it make some inflammation caused by far-UVC after 5 seconds. And this is the analysis of apoptosis in the cornea epithelium through the tunnel assay at different seconds after far-UVC to 2 nanometer exposure. So here we also got it the data, like if we give exposure more than 5 seconds, so we got it some signals on the first layer of the cornea epithelium on 10 second and 15 second on 20 second. Even in 20, after more than 20 second exposure, it detached the epithelium, so it's very strong exposure. So this is the another figure in which we just check the we just counted the colonies after far-UVC exposure at different time interval. So here we also get the data, like if we give exposure for 3 seconds, it will be more effective compared with others. This is the another picture in which we check the statistic analysis comparison between normal iodine and UVC. So here we can see that the UVC have very good result compared with iodine and normal one, and here is also a significant figure also shows that UVC is much more effective compared with iodine group. And here we also got it some bacteria in each strain after giving exposure. This is the different types of bacteria which we got it on each cornea. This is another figure in which we check the inflammation in the stroma through ROS assay. The conclusion of this figure is only like if we use iodine for a long time, so it will be penetrated through the cornea epithelium layer, and it will make some inflammation in the stroma. So we mean that it's not better to use the iodine. And this is another to check apoptosis. So this also give us idea, like if we use iodine, so it also makes some DNA damage which is got it by we check by the tunnel assay. And this is the conclusion of my progress report and their application in clinical ophthalmology. We already found that if we use the 9 millijoule per centimeter square of far-UVC to 2 nanometer exposure, so it will not cause any harm to the cornea epithelium cells or stroma, and eliminated bacteria as effective as iodine. According to our study, if we use this 9 millijoule per centimeter square of far-UVC to 2 nanometer exposure is safer and no more effective for disinfecting ocular surface for ophthalmic surgeries than iodine. So this result supports the idea of far-UVC to 2 nanometer disinfectant providing safety, efficiency and lower cost for clinical ophthalmology. So this is the future study direction. We already got the data like far-UVC to 2 nanometer light have the ability to kill the bacteria or any type of microorganism on the surface of cornea. So why not we can use this far-UVC for bacterial keratitis, which is a disease of eye. Earlier I explained in my second semester progress report. So this is a study design in which I will induce bacteria in different groups and then I will check their effect with each other. This is a time point which I give drugs to my rats from 8 o'clock at morning to 10 o'clock at night, and after 7 days I will sacrifice them and I will check H&E staining, tunnel assay, ROS assay and PS staining. And the summary of my future this study, we just want that far-UVC to 2 nanometer light exposure therapy and induced bacteriophage will be a new effective way of treatment for bacterial keratitis. And also the point like still like there is drugs is used, antibacterial drugs which is used for bacterial keratitis, that type of diseases, but still some bacterial drug resistance which are not killed by drugs. So we hope that maybe the bacteriophage model and far-UVC to 2 nanometer light exposure will be a new effective of treatment for bacterial keratitis. So our purpose from this study is only generated the new way of treatment for bacterial keratitis and will be a lower cost in future. Thank you for watching.